All right, we will talk about carbohydrate digestion, then its absorption. As I mentioned, carbohydrate digestion begins in the mouth with the help of salivary amylase. This enzyme acts on complex carbohydrates. You're thinking about polysaccharides, and when polysaccharides are digested in the mouth with the help of salivary amylase, they're broken down to they're broken down partially. So you're talking about oligosaccharides. So they're not as large as polysaccharides, but they're not very small either. So they're in between. Or I may say disaccharides. Disaccharides are two glucose molecules or two sugar molecules in general. So it's glucose fructose or glucose galactose and so on. Okay. So that's in the mouth. Now in the stomach, esophagus, is where you don't have any enzymes, but the enzymes that are acting on these sugars continue to be acted on a little longer as they travel through the esophagus. However, once it reaches the stomach, something happens. That is, the acidic nature will make salivary amylase not function anymore. Acidic nature that would affect the action of salivary amylase, inhibit the action of salivary amylase. You see? So no further digestion in the stomach. Let's go to the small intestine. This is where we have a pancreatic amylase. Pancreatic amylase would act on the oligosaccharides, disaccharides, and break them into more disaccharides, and those disaccharides will now be monosaccharides. So this is a combination of disaccharides and monosaccharides. This is through the help of pancreatic amylase. All right. So once we are at this stage, we have to note that there are enzymes on the surface of these brush border cells. And brush border cells are epithelial cells with the microvilli. And these enzymes are sucrase, maltase, and lactase. All right, they are on the brush border surface. So they digest these disaccharides further into monosaccharides. Monosaccharides, we would come up with 3 of these. That is glucose, glucose, fructose, and galactose. All right. Fructose and galactose would eventually be converted into glucose in the liver. All right. So this is all about digestion of complex sugars into simple sugars. These three are called simple sugars. Now let's talk about their absorption. All right. You have brush border cell here, and then brush border cells here. So this is where you have the monosaccharides. That are now going to be brought into the cell. This is brought in by a mechanism that you define as co-transport mechanism. So that is glucose along with sodium ion. Okay, sodium ion. So now glucose and sodium ion together would be arriving here, and then facilitated diffusion is how it would reach the blood capillary. Okay, so you're talking about blood capillary. Eventually it would go into the blood vessel. So glucose is absorbed. So this is the absorption of glucose from the lumen of the digestive tract here all the way into blood. All right, with this, let's talk about protein digestion. Protein digestion starts in the stomach. The enzyme that you need to be thinking about is pepsinogen. All right, so you're talking about pepsinogen, an inactive enzyme made by the chief cells of the stomach lining, and pepsinogen is converted to pepsin with the help of hydrochloric acid. You see, so once pepsin is released, it's an active enzyme, so it would now convert proteins. Okay, pepsin would convert protein or a polypeptide to peptide. Okay, so basically what you're thinking about is protein being a very long structure, or a polypeptide, a long chain of amino acids would be broken down into a peptide. Okay, so it's not too short, not too long, somewhere in the middle. So pepsin is the one that does that in the stomach. And then when we go to the small intestine, that's where it's important for us to know the list of pancreatic enzymes that are involved in the digestion of proteins. Well, let's start with trypsinogen. Trypsinogen, this is a pancreatic enzyme, all right, that is going to be released into the small intestine. And in the small intestine, you have enterokinase that converts it into trypsin. You see? So once you have activated trypsin, trypsin would in turn convert another inactive enzyme, chymotrypsinogen. Chymotrypsinogen will be converted to chymotrypsin. So these two are the two key protein digesting enzymes. They would act on any remaining polypeptides, proteins to peptides. So this is in the small intestine. The enzymes are pancreatic enzymes. Chymotrypsinogen is also a pancreatic enzyme. Okay. Anytime you see O-gen, it's an inactive enzyme. Please note that the reason why they are released in their inactive form is to make sure that they do not digest the cell itself. All right. So now let's go to talk about what happens next. So now we have peptides that are short chain of amino acids. As long as they're dipeptides or tripeptides, di or tripeptides, then there is an enzyme called peptidase. Okay. So peptides are again broken down by trypsin, chymotrypsin, you know. So tripeptides, peptidase would convert it into amino acids. Where is peptidase? Brush border cell. Got it? So amino acids now absorbed by sodium amino acid co-transport and facilitated diffusion to blood. Okay, so you know how it would be brush border cell here. This is where it's sodium co-transport, facilitated diffusion here. And that's how you would have amino acids brought to blood. And that's what you call as absorption. Okay, good. With this, we can now move on to talk about lipids. Okay, lipid. First, we'll talk about digestion of lipids. Well, because they're large molecules, you need emulsification first. So the lingual lipase in the mouth, gastric lipase in the stomach. All right. They both, lingual lipase, gastric lipase, they both are not as effective. Why? Because emulsification doesn't happen until lipids are in the small intestine. So let's talk about what happens in the small intestine. Small intestine is where you have bile. All right, bile salts are the ones that convert large globules of lipids into multiple small fragments. Okay, so this is a soap or a detergent action. Okay, And you simply call this as emulsification. And this is where you have increased the surface area. Now, the enzyme that you're thinking about is pancreatic lipase that would break the lipid into monoglyceride and fatty acids. Okay, so the lipids are now converted into monoglycerides and fatty acids. All right, once you have that, you have to transport somehow, all right, because they're not water soluble. So protein, all right, these are plasma proteins, protein plus monoglyceride plus fatty acid is what you create, and all of this together is what you call as a micelle. All right, so this is what is created next. Micelle is going to transport monoglyceride and fatty acid to the brush border cell. All right, so it brings this structure to the brush border cell. So fatty acid and monoglyceride are brought into the brush border cell. The protein would go back to pick up more of the same, basically shuttling back and forth. The strange thing that happens in the brush border cell is these two come together and become a triglyceride. Triglyceride, a large lipid, is formed, okay? And now we need to transport it out of the cell, so you need to add a protein to it. So here you create this large structure that you refer to as chylomicron. Chylomicron. It's a protein lipid mixture, very big. So now we have to think about a way to transport blood capillaries too small for it. So you move this structure into a lymph capillary. All right, chylomicron here. Okay, lymph capillary that transports chylomicron in large amounts would be called as a lacteal, milk-colored vessel. Okay, chylomicron, as it travels along the lymphatic veins and ultimately blood, it ultimately joins blood circulation. As it travels through the blood, there are enzymes called lipoprotein lipase. Lipoprotein lipase releases fatty acids that are either taken by the cells or stored in the adipose tissue. All right, so chylomicron is now used in the release of fatty acids that could be taken by the cells of the adipose tissue. Any leftover chylomicron is called chylomicron remnant reaches the liver, where liver uses it to synthesize new lipids and release it back into blood. New lipids are called very low density lipoprotein, VLDL. And that would be converted into intermediate density lipoprotein. Ideal intermediate density lipoproteins. So this is again the action of lipoprotein lipase, LPL. All right, ideal can be taken back to the liver for storage. Ideal can be converted to low density lipoprotein, LDL, by lipoprotein lipase action. Okay, LDL is the one that would be taken by cells for energy purpose. So this is smaller, can make its way into the cells, whereas the others are still big. So this is how lipids are transported and utilized by the cells. So good. We'll stop at this and pick up the large intestine detail elsewhere. Thank you.